is collaborative work with the New York State Institute for Basic Research, and we have Dr. Fan and Anne also online, and so that's wonderful. This is a continuation of work from a different for many years and different collaborations that has evolved into a very solid framework that Richa has further developed and will present to us today. Once she's, I also have to mention that once she's done with all the paperwork and so forth, we're going to be very sad to see her go to a postdoctoral position in Chicago at Northwestern University at the Institute for Rehabilitation at Chicago. Rehabilitation Institute of Chicago, Rehabilitation Institute, right?
Yeah, the Ability Lab.
Ability Lab, which is a large, very well-funded, top #1 place for rehabilitation in the US. So she's going to be very productive there and hopefully build bridges that will continue to interact. Otherwise, we're going to miss her terribly. It basically keeps the lab glued together. Amazing work. So I look forward to your presentation and go for it.
Thank you, Liz, and thank you everyone for attending my defense from heart rhythms to scheduled motor milestones, a novel approach to early stage neurodevelopmental monitoring. So to start off, I'd like to give an overview of how we look at development, specifically infant development. We view it under the lens of the dynamic system theory, which essentially says that typical neurodevelopment requires increasingly complex systems in order to mature, and that infants use these systems to produce new ways of exploring and controlling their environments. So these systems can refer to our perception or various other parts of our sensory system, or the countless parts of our motor system, but they all must work together and develop properly in order for a child to exhibit what we deem as typical behaviors. So when we view behavior as a very complex and variable thing that is always changing based on the internal and external environments of the infant, and this theory is based on mathematical models that describe time-based systems with complex and nonlinear dynamics, like development during infancy. So we know that behavior and social emotional factors emerge as a result of the multiple components of the complex systems at play. And we theorize that behavior is the product of multiple contributing influences. And in order for us to see what we deem as typical behaviors, all of these subsystems need to develop together. So at this point, you might ask, if we're looking at infants, why are behaviors already so important? And this is because currently neurodevelopmental disorders like autism, the rates for which are growing quite rapidly, are diagnosed almost entirely based on behavioral paradigms. So what this means is that a child is diagnosed based on specific behaviors they present, and based on what is considered atypical, they will get this diagnosis. The average age for diagnosis right now is about 4.3 years old. And by this age, many of the complex systems which are developed during infancy and are responsible for behaviors shown at around this age are already in place. When looking at the structure of systems, things like our motor control develop very early on. And these systems, which also do show atypicality at earlier ages, would serve as better markers for the tracking of neurodivergent development. So they could let us catch disorders earlier and help provide more appropriate therapies in a more timely manner. So the goal of this work and of our lab is to work hand in hand with the clinicians who are currently the experts in this field to develop objective metrics of tracking and diagnosis for developmental disorders and to detect atypicality in development. So ideally, we can take already well-established and well-proven paradigms used for tracking development and work to digitize and adapt them to a system that can provide clinicians with more objective metrics in a way, ideally, that is not burdensome for the clinician or for the patient. So as an example of one of these tests is the general movement assessment. It's a non-invasive clinical assessment that can provide information on how a baby's neurological system is developing based on their movement patterns. So it's given normally when there are medical concerns at birth or if a doctor sees any abnormalities. So it's not something done with every single infant, but the purpose of it is to capitalize on an infant's neuroplasticity so that the clinician can intervene early on, especially if there is some diagnosis or an abnormality. In general, it's very reliable. However, it needs to be done with a trained professional, and it's not done with every baby. So those who don't present any atypicalities will not be evaluated, and well-trained practitioners are not easily accessible for people in different areas with different socioeconomic conditions. So ideally, if we are able to digitize something like this and make it more accessible to parents and to doctors who are potentially not trained in it, we could work to detect and intervene for infants in a more reliable way. So we believe that this is possible because we have found that in a previous study done by the lab, we can detect neurodevelopmental derailment by 3 months of age using data from a wearable sensor fixed to the leg of an infant. In this work, about 9 to 12 hours of data was collected once a month over the course of 5 months in 60 neonates. And by 3 months of age, they were able to isolate infants who had stunted growth or stunted development of neuromotor control. These results were later validated when several of the flagged infants were later diagnosed with autism in their early adolescence. The work also found that the development of motor control was not linear, and when using metrics like the standard growth chart, variations can be overlooked. So we want to add to this by, one, making it more accessible, since research grade wearable sensors and data analysis techniques are not something everyone can have access to. And we want to study the environment and the infant, which we will do so through the evaluation of dyadic interactions. Another important part of an infant's development is their environment, specifically how an infant explores and interacts with their environment, especially with their caregivers. Dyadic interactions are extremely integral to our social life and are a foundational building block of social interactions. There is a lot of work which supports that a parent's involvement in early childhood years is quite formative, so we also believe it is important to study the dyadic relationships in an infant's life, since this could provide another lens to view their development and track their growth, as we can measure the changes in these dynamics over time. Specifically in this work, we will be looking at physiological relationships between mothers and their infants, and with several infants and a clinician. An overarching goal that this work hopes to contribute to is the development of a digital growth chart. Currently, pediatricians use body weight, body length, and head circumference to match babies to a physical developmental growth trajectory, as seen here, to see if development is progressing at a typical rate. We propose taking this to the next level and charting development based on various objective physiological signals. Ideally, this will be an accessible and scalable metric that can be used more accurately and objectively to track development, and therefore more accurately catch atypicalities in development. To do this, however, we need to track many infants longitudinally. So in this work, we are proposing methods that can be used to objectively and digitally track the schedule of maturation in infants. This entire framework is grounded in a personalized medicine approach, which is a multi-layered approach to the development of a personalized and targeted treatment. It relies on the proper integration of all of the layers in the network here. At the bottom, we have clinical inventories relying on observation of behaviors, which is the predominant practice in this field today. We want to connect these layers with digital phenotyping and develop digital biomarkers that are informed by these clinical criteria to then progress toward integrating all of these layers and eventually work toward more precise, personalized approaches for therapeutics and diagnostics, which would lead to earlier interventions. So these are proven to be more helpful than interventions that take place later in life, since earlier in infancy, our systems are again more plastic, and the development of some of our complex systems are reliant on underlying mechanisms. And if one of those underlying mechanisms is not developed in a proper way, systems dependent on that scaffolding may suffer. All of the studies in this dissertation utilize the precision of a unified analytic framework to quantify the underpinnings of the critical developmental phase that takes place during infancy. It has provided to be a very valuable tool in studying various types of biosignals in our lab, So I'm going to go over the general principles now since they are applied throughout the rest of the presentation. Crucial to this framework are the micro-movement spikes derived by the formula seen here in panel B. These are normalized values which range between 0 and 1, and they represent the intensity of deviations from the empirically estimated mean of the signal. The beauty of these spikes is that they are normalized according to your own baseline, so they account for allometric differences that are present across individuals. So they serve as a very good metric when studying cohorts of people. They also serve as a good metric for longitudinally studying infants who are going through very rapid physical changes. So these normalized peak values from the micromovement spikes are then used to plot a frequency histogram as shown here. We can then empirically fit a probability distribution function using the maximum likelihood estimation, which has shown to be the gamma family of probability distributions. The gamma family has shown to be the empirical distribution for human biorhythmic data in the lab, and we've seen this can be the case for various types of movement. So we can plot the shape and scale of this gamma function, which has been empirically estimated to fit the data with a 95% confidence interval. Here you can see several windows plotted, and this blue point refers to the specific function. The scale refers to the noise to signal ratio. We will typically use it as a metric for noise, and it is derived as seen here. The shape is the inverse of this and is a reflection of the shape of the distribution. The lab has discovered that the log-log of the gamma estimation yields a tight linear fit, and that when the noise reduces, the shape increases to a more Gaussian shape, which is more symmetric. This tight linear relation is then used to track the points within the parameter plane, which we can follow over time of neurodevelopment. So to better understand this plane, we obtain the median values of the trajectory, which you can see in the median lines on the rightmost plot, so the median shape and the median scale, and we divide the plane into 4 quadrants as seen here. So you can see all these markers, which represent values from different windows from different PDFs seen in the first panel, all from one single time signal. This division brings us the left upper quadrant and the right lower quadrant, which we track throughout this work. So the left upper quadrant contains the instances that have high noise and low shape value. This reflects a random and memoryless state, and prior research has found that states of autism and neurodevelopmental disorders predominantly remain here. The right lower quadrant reflects probability functions that are more toward a Gaussian range with lower noise. So these instances more so reflect a learning process seen in more neurotypical developmental states, and ideally in a healthy learning system, we will see transitions between these quadrants. However, if the regime stays in one quadrant, we tend to see neurodevelopmental differences. So what we want to remember is that the left upper quadrant reflects a state of more randomness, similar to the early exploratory phases seen in infant movement, whereas the right lower quadrant is a transition to more intentional and predictable movements seen in typical babies. And it is important to remember that these instances are not static, so there is movement between quadrants. So we need a way to measure the directionality and the magnitude of change over time. To do this, we utilize the Earth mover's distance. This is basically a metric used to quantify the amount of effort required to transition from one state to another. If we take these 2 histograms here, the Earth mover's distance can give us a metric that quantifies how much change it would take to transition between the 2. So we can see in panel B a representation of the Earth mover's distance over 9 windows from one recording, and we can track this metric longitudinally, and from it we derive what we call critical points. So these are the maximum Earth mover's distance values for every instance we are studying, and you can see here that we plot these critical points with the marker representing the quadrant in which they exist. So this is a way for us to visualize the trajectory of each infant's development over time. So in total, this dissertation consists of 3 different studies. In the first, we look at heart rate variability data in a dyadic setting between a mother and her baby. In the second, we look at longitudinal and cross-sectional data derived from video-based pose estimation of infants. And in the third, we use wearable sensors to track the motor development of infants using the clinician as a frame of reference. And we work to validate the accuracy of pose estimation data. So diving into study 1, we are going to introduce new methods for the monitoring of mother-infant heart rate interactions within the context of their daily life. The overarching goal here is to chart the trajectory of the dyadic cardiac interplay that will unfold organically over the course of the day. So heart rate variability is characterized by the fluctuation of the time intervals between consecutive heartbeats. Here you can see that after we filter the raw heart signal data, we get these peaks called R peaks, which signify each heartbeat in the data. So the interbeat interval, which is the timing between each heartbeat, is what we use to derive the micromovement spikes. HRV is an entirely non-invasive measure, and it represents a functioning of the autonomic nervous system's dynamics, specifically the interplay between the sympathetic and the parasympathetic systems, and its regulation of overall cardiac function. It reflects the heart's ability to respond to a variety of stimuli, both environmental and psychological, which makes it a very good measure for studying dyadic interactions because it can inform us of how the heart is adapting to changing internal and external demands. There is a host of literature that talks about the biobehavioral adjustments that take place in infancy as infants adapt to the early weeks of life, specifically about the strong relationships between mothers and their infants. We've seen that mechanisms like skin-to-skin contact, which is recommended by doctors immediately after birth, and paced breathing of the mother can have a calming effect on the infant and help synchronize their systems. So in this study, we went in wanting to see if a mother's influence of the autonomic functioning of an infant could provide any insights to their overall functioning and their developmental trajectories. And although heart rate may seem like a general measure, it's been shown to have a predictive power with regards to an infant's adaptability to stress and their overall resilience. So we want to see if this physiological regulation might lay the foundation for later developmental achievements through our new parameterizations of HRV. The data used in this study was given to us by Dr. Sam Wass at the University of East London, who was able to share unidentified electrocardiogram data, or ECG data, for 18 mother-infant pairs. In this study, all of the babies were 12 months old and their mothers were the primary caregivers. To collect the data, a researcher went to the homes of the participants and set up a specially designed ECG system on the mother and the infant that you can see here. The mom had a band wrapped around her chest, and the baby had a bodysuit that had the sensor embedded. So these sensors were able to have direct contact with the skin, and the participants had full mobility throughout the recording. The researcher then left, and the mother and baby went about their day while the sensors recorded their heart rate activity. The data we got was anywhere from 3 to 8 hours in length. However, the issue for us was that we were not provided with the labels for this data. So the group had audio and motion recordings that could not be shared with us. So we went in entirely blind when analyzing, meaning we had no idea what was happening or what the mother and child were doing at any point during the recordings. This adds another layer of complexity to our work because without labeled data, we have to instead rely on already established methods of validating whether or not the data is usable. To validate whether our filtered signals were, 1, from healthy individuals, and 2, were reliable cardiac signatures, we utilized 2 well-established methods in the field of HRV analysis. The first is detrended fluctuation analysis, or DFA, which investigates the temporal structure of the signal, and it assesses the dynamics of the autonomic nervous system. It can differentiate between short-term and long-term fluctuations, and it's important because it can evaluate based on separating random fluctuations from variations that could be the result of other physiological processes. It has also served as a prognostic tool and has been used to identify cardiovascular dysfunction amongst other abnormalities. So it has established that in a healthy signal, its exponent will have a value of about 1, which all of our subjects in our study had. So we use this as a way to validate that the participants were indeed healthy and that the data was processed and cleaned effectively. On the right are Poincaré maps, which are scattergrams that are a valuable tool when analyzing the temporal information of heart rate data. They are traditionally a visual technique that can help analyze dynamic systems. What you see here is each IBI plotted as a function against the previous IBI interval. So normal heart rate signatures are typically defined as a symmetric fan-shaped scatter, as seen here, and this was seen in all of the dyads we studied. These maps can also be looked at quantitatively. We can fit an ellipse that you see here to the scatter, and this helps assess the sympathetic and parasympathetic activations of the nervous system. As a very brief explanation, the sympathetic nervous system is what is responsible for our fight or flight responses and prepares our body for stress and emergency, whereas the parasympathetic is responsible for rest, and together they contribute to the functioning of the autonomic nervous system that regulates our heart rate. The minor SD1 axis here is a reflection of the parasympathetic system, and the major axis, the SD2 parameter, is a reflection of the sympathetic system. The ratio of these two, SD1 over SD2, is another important parameter we use, which represents the ratio of short interval to long interval variation, which basically means that it represents the randomness of the heart rate variability time series. So this is often used as an index of variability between heartbeats, and it's informative of the equilibrium in the autonomic nervous system. We use the unified analytic framework discussed earlier to analyze the micromovement spikes derived from the IBI time signal. So we're able to track the stochastic evolution of the scale or the noise by tracking the occurrences in both quadrants. In the third panel here, you can see that we plot the scale over time, and the markers represent which quadrant those instances occurred in. We then take the time location of the instances for the LUQ and the RLQ of mom and baby and put those in histograms. We then take the earth mover's distance of all of the histograms from the mom and baby for both quadrants. So the right EMD matrix here is a comparison of all the mom and baby occurrences in the right lower quadrant, which again is where we see more predictable signals, whereas the left is from the left lower quadrant, which again is the memoryless and more random quadrant. Another metric we use here is transfer entropy, which is an information measure that quantifies the coherence between two systems. It's able to essentially distinguish between elements that drive change versus those that respond to change, and it can quantify the degree of influence between these systems. So essentially, when applying it in this scenario, we can see who is influencing whose heart rate, mom or baby, and to what degree. What you see here on the bottom left are the transfer entropy matrices for the SD1 over SD2 ratio for the baby's influence on the mom, and next to it, the mom's influence on the baby. We've divided it into 3 separate time domains, signifying earlier in the day, middle of the day, and later in the day, so you can see the degree of influence throughout these 3 instances. For example, if we compare this top row, which is our first mom-baby dyad, you can see that in the very first rectangle, the baby to mom influence is a lighter color, which means it's a higher value. So the baby has a higher influence on the mom's ratio or overall variability of the heart signal than the mom does on the baby early in the day. Overall, lower values here are indicative of a more static system state, and higher values suggest a more dynamic exchange. Next, we plotted the median noise to signal ratio for mothers against infants for each of the plots in the quadrant, and we found that there were 2 self-emerging clusters in this visualization, which is understandable since we plotted the left upper quadrant and right lower quadrant. We used k-means clustering to cluster all of the data points, and this validated that all of the points in cluster 1 are from the left upper quadrant and have a higher noise to signal ratio and lower shape parameters. And in contrast, cluster 2 contained all of the points within the right lower quadrant with lower noise and higher shape parameters. It's obvious that these quadrants will cluster again, but k-means gives us a centroid, which we can then use as a reference to detect extreme cases on this plane. The color of the markers here are the log of the earth mover's distance amongst all of the dyads, so it tells us the similarity amongst all the pairs, the bluer color showing a higher degree of similarity and the yellow showing a lower degree. So using this stratification, we decided to dive deeper into these clusters to study the nuance between the points in each. So we further divided each primary cluster into subclusters based on their vertical positioning within the noise to signal ratio space, which has given us 4 distinct groups: the extrema of the left upper quadrant in blue, extrema of the right lower quadrant in red, and 2 divisions in the middle in tan split in half. This division will allow us for a more granular examination of the dyads, and today we will be focusing specifically on the top and bottom halves of each of the clusters, since empirically these are the most distinct. So here we can see 2 dyads from the right lower quadrant, one where the baby leads and the other where the mother leads. Each marker here represents a 1-minute time window from the original recording, and the plots here show the relationship between the noise to signal ratio and the SD1 and SD2 metrics. The color denotes the temporal sequence throughout the day, so the darker blues represent points from earlier in the day, and the yellows points from later in the day. We see that the infant spaces have more of a concentration of specific colors in specific areas of the plots, which suggests a more stable physiological state over the course of the day, whereas the mothers exhibit a greater scatter, which could indicate a wider variation in autonomic activity during the same time period. This could be indicative of a more heterogeneous response to daily stimuli, while the infant scatter implies a more controlled state during the day. When the baby leads, the mother's dispersion is a bit more controlled, and we see that when the mom leads, it doesn't have a large impact on the dispersion of the baby since the scatter is more contained. Here we see two dyads from the right lower quadrant again, one where the baby leads and the other where the mother leads. We again see that the infant space suggests a more stable physiological state. However, here we see a general greater dispersion in the baby scatter when they are leading and a more controlled when the mom is less regulated, which again shows that the mother's autonomic state doesn't directly influence the infants. So these scatters are a better representation of the transfer entropy throughout the day. Using the SD1 over SD2 ratios as the primary metrics, each plot shows us who is leading the leading party based on the axis highlighted in red. As we see in the transfer entropy matrices, the color within the scatter corresponds to the time of day that the leading party had the most effect on the other system. From this, we saw overall that infants more frequently exert a dominant influence on the autonomic system of the mother, and in general, mother's autonomic states were shown to be less influential. The time of day didn't seem to impact this general pattern, and the unity line is a reference for what we believe would be a balanced autonomic state, so points hugging this line would suggest an equilibrium between the mother and infant. What we see here on the top row is the SD1 over SD2 curve of the baby and mother superimposed. Again, this ratio informs us of the overall variability of the heart rate signal. So the area between these curves can inform us to the overall similarity between the variabilities of all the pairs. So you can see the normalized values, which provides us with a metric of what pairs are most to least similar. We also look at the extreme cases of the noise regimes. And in this figure on the left, we see the day-long trajectories of the absolute difference of the SD1 over SD2 ratios, which contrasts the noise levels between the mother and the infant. We see here that the baby signature is impervious to the mother's increased dispersion, and that higher variability of mom does not directly influence the baby signal. In the right figure, we extended these findings to density distributions of the absolute differences of the SD1 over SD2 ratios. When the baby has a low dispersion, there is a high signal transmission to the mom, and overall, the mom's signal seems to be noisier. So this could suggest that the mother's autonomic state is potentially more reactive or more sensitive to higher physiological noise levels. And overall, it again shows us that the infant tends to have a stronger influence on the mother's signal. So overall, in this chapter, we found that from an entirely blind data set, we can find self-emerging clusters of the interbeat interval gamma-based noise signal, and that we can track these dynamics throughout the day. Minute-by-minute segments reveal the exchange of information from our transfer entropy analysis, and most significantly, we saw that the overall influence is an infant-led influence on mother HRV, which could signify a more responsive maternal autonomic state. This is quite significant because generally in this line of research, the mother is seen as the base and the child's reactions are in response to her regulation as it's monitored. So it's interesting to see that the mother is also sensitive to the baby's autonomic regulations. With more research, we could further explore this transfer information to understand causal mechanisms at play under these patterns. Longitudinally, we could track these dyadic interactions over more time, which could potentially provide us with more insight into long-term developmental impacts of a mother and child. Now we are going to shift to study 2, and for the next 2 studies, we will be focusing more on the development of motor control. For this study, we will be proposing several new techniques for the longitudinal assessment of video-based tracking of infants and their movements. The purpose here is to eventually combine this information with traditional methods, such as those used to measure physical rates of growth, as we saw in the standard growth chart in the introduction. Our overall goal here is to develop personalized assessments tailored to accurate rates of development of each infant that can be followed with the infant's individual maturation schedule. So clinicians often use babies' very early motor patterns to gauge and predict the integrity of neurological systems. One very established test that we chose to study here is Prechtel's general movement assessment. This test relies on the subjective observations by a well-trained clinician who uses standardized criteria to detect typical and atypical movements. The GMA has successfully identified abnormalities in neurological development and growth and has been very successful in detecting cerebral palsy during infancy. The test focuses on the observation of movements, predominantly of the limbs, and it divides the first 30 weeks of life into periods of writhing, fidgety, and intentional movements. The writhing movements, present from about 0 to 6 weeks, are described as small swimming-like movements with moderate amplitude. They appear from birth and then transition into fidgety movements, which are more variable and have a moderate speed, and more so involve the neck and trunk while the limbs move in all directions. We see this until about 15 weeks, after which we expect a transition to intentional movements, which are more controlled and less variable. Clinicians are trained to observe the pattern in these movements, and they can detect when something is abnormal or if an infant isn't progressing to the next stage at an appropriate time. However, this test is only given when there is either belief of some developmental delay, if there are some complications during birth, or if there are genetic predispositions that need to be monitored. So not all infants are tracked in this way. It also requires frequent visits so that progression can be tracked over time, which is difficult to do. We believe that using technology to track biorhythmic motions from birth could offer a higher level of precision to complement these clinical criteria, and could also serve as a new way to understand how motor organization develops and helps us understand the beginnings of neuromotor control. We believe that by digitizing the GMA and later on other clinical criteria, we can create more objective metrics of detection that are scalable and usable in various populations, and that we can help clinicians in the detection of abnormal movements that are a function of the neurological system. So to track these movements, we've used the foundation of the GMA, which identifies the proximal group of body parts, such as the head, neck, shoulders, and the parts closer to the center of the body, and the distal, which are the distal limbs and end effectors. However, we add a new classification of articulated joints, which are the joints that connect the limb segments in the arms and legs, so our elbows and knees. We know that the proximal class stabilizes first, since they provide the foundation for the distal class, such as the wrists and hands, which the infant uses predominantly to interact with and explore their environment when they are mature enough. To track these movements, we developed an app, Baby Motion Lab, which parents use to submit videos directly to us. So they would select the position which they would like to record their infant, and then they were able to submit a 1-minute, 30-second video that was able to be sent directly to the lab. We then analyzed these videos later on, and they were submitted for about 16 to 20 weeks over the course of the very early weeks of the infant's life. Once we had these videos, we used OpenPose, which is an open source pose estimation toolkit from Carnegie Mellon, to extract the key points of the infant. The key points refer to the little dots that you see on the colorful skeleton of the baby, and these are the points being tracked. So OpenPose will return a time series which contains coordinates for each of these points, which we can then analyze. We worked with the pose estimation data for all of the videos submitted, which provided us with the longitudinal data set for each of the 5 infants in this study. So here you can see the raw kinematic pixel per frame data that we derived from the output of OpenPose. This represents the physical kinematic speed of the infant's movements. In this chapter, we look at the head, neck, both shoulders and both wrists, and we set a confidence threshold of 75%, which means that we only looked at data points that the software believed to be accurate with 75% certainty. Since this model is not trained specifically on infants, we weren't confident in tracking anything below the certainty, which happened to be most of the lower body. We selected these 6 points because they give us a good representation of the upper body overall, and they provide us with 2 body parts for each class, so the head and neck in the proximal, the elbows in the articulated, and the wrists in the distal. So the first metric we will be looking at is the physical pixel units. We are able to convert the distance of pixels to physical millimeters based on the phone's recording configurations. So we were able to track the physical movement of the infant, which means that we can get a tangible measure of the infant's total physical movement per session, and then use this to track the movement longitudinally over the course of the week studied. The cumulative sum metric that you see here was calculated for each week, and it represents the total sum of movement for the specific day and all the days below, which can give us a view of the infant's physical speed over time. In panel C, you see the cumulative sum superimposed with the polynomial line of best fit, which can help us underscore the overall trend of increase of motor activity, and it gives us a quantifiable metric for the rate of development of the speed of each body part. We would expect to see increased speeds as the baby grows, and therefore a steep slope, because this would show change which we would want to see in early weeks. If we saw a flat slope, we would then flag that there is stagnation early on. So here you can see the raw kinematic pixel per frame data that we derive from the output of OpenPose. This represents the physical kinematic speed of the infant's movements. In this chapter, we look at the head, neck, both shoulders, and both wrists. We set a confidence threshold, as discussed earlier, 75%. And this model gave us that these data points were all confident within 75% certainty. Since it's specifically not trained on infants, we weren't very confident in tracking anything below the certainty, which happened to be most of the lower body. We selected these 6 points because they give us a good representation of the upper body overall, and they provide us with 2 body parts for each class, so head and neck for proximal, elbows for articulated, and wrists in the distal. So here you can see the superimposed cumulative sum trends for each baby over time. We've divided the plots into writhing, fidgety, and intentional movements to match this maturation to the established clinical paradigms. The trajectories we see establish a clear developmental pattern across all of the infants with distinct variations in body parts. For the proximal body parts, there is a steady increase in cumulative movement with minimal variability between babies, which can suggest a foundational level of motor stability in the head and neck regions consistent across the cohort. This makes sense since this area plays a large role in providing stability. In the articulated body parts, we see a more pronounced inter-individual variability, particularly where the highest cumulative movement is observed in later weeks, which reflects a broader range of movement in the elbows. In the distal body parts, we see an even more pronounced inter-individual variability, and we see the largest overall movement, so we can view this region as having the most rapid scale of motor development. This reflects a broader range of movement, exploration in the limbs compared to the head and neck, and overall we see that the distal joints have the steepest slopes, followed by the articulated, and then the proximal, which may reflect a developmental progression of the stability of movements. From the slope data, we can see that there are differences within the body parts of specific babies. For example, if we look at baby 1, the slope data suggests that the wrists exhibit a steeper increase in movement over time compared to the elbows, which have a steeper slope than the head and neck, which quantifies that baby 1 is potentially exploring more with the wrists than anything else. We can also look at symmetry. So if we look at baby 4, we can see that the right wrist has a steeper slope from the left wrist, and the right elbow a steeper slope from the left elbow, which indicates overall more activity on the right side. So this could potentially represent a preference in movement on that side, or it could be related to the infant's environment and their responses. Also, when looking at the differences between individual infants, we saw that in general, right-sided body parts exhibit a steeper slope than their left-sided counterparts, which could imply a side-specific maturation pattern that we can explore with more investigation. Here we can also see the average sum per week. In the proximal region, we see a stable pattern across all of the periods, with one baby showing a bit of variability in the writhing period. In the articulated class, we see that there is variability in the writhing, and then there is stability throughout. And we see that in the fidgeting period, it is a transitionary stabilizing period because we can see variability in the writhing and intentional periods. In the distal class, we see variability during all the periods, which reaffirms what we found earlier, that the distal class is the exploratory mechanism of the baby, and that it may rely on the articulated class to enhance these exploratory patterns. After running statistical analyses using the non-parametric ANOVA-Kruskal-Wallis test and an assessment via the Wilcoxon-Ransom test, we found that in the proximal and distal groups, the writhing phase was significantly different from the fidgety and intentional phases. And in the articulated group, there was a statistical disparity between the writhing and intentional groups, but not with the fidgeting. So this suggests a stable transition before the shift in motor control, and all of these results were statistically significant. So now we are going to give a brief overview of our cross-sectional analysis, where we group all of the data from all the infants that we recorded into different age cohorts to map the trends within each cohort over time. So we divided the first 18 weeks into 3-week cohorts and then evaluated all the recordings that occurred during those time periods. We took the Earth movers distance of all those recordings as we previously discussed, and we found a range of the maximum and minimum EMD values across all the cohorts, the maximum being the solid line and the minimum the dotted. So when looking at the max EMD, we can see the range of the dynamic shifts is very large in early weeks for the proximal and articulated joints, followed by a stabilization toward a narrower range. We also see this pattern in the right wrist. The importance of tracking these values is that they can help us understand boundaries of motor development variability if we work toward having a larger sample size and a more representative patient population. So overall, our analysis of the cohort reveals that while individual infants do display distinct motor trajectories, we do see the emergence of a common developmental pattern. There is a discernible sequence in the maturation of motor control progressing from proximal to articulated to distal joints, and the data suggests that as infants grow, the use of their distal joints increases, reflecting the natural progression of motor skill acquisition and setting the foundation for later cognitive development. Distal joints are the most relevant in allowing the infant to explore their environments, and these findings do corroborate clinical methods. We were also able to find distinct transitions within the established writhing, fidgety, and intentional periods. More digital characterizations here could be pivotal in targeted metrics for detection and evaluation of progress during different therapies. The main premise of this study was to find a reliable, accessible, and convenient form for early detection during the early weeks of infancy, specifically to develop a tool and methods that clinicians could use in the office or could provide to parents in the home to digitize already existing methods. So now moving on to our third and final study, where we use both wearable sensors and pose estimation data to gain an even deeper understanding of the development of motor control during the early weeks of life. Here we will look at different poses clinicians use and how to digitize them while considering signals from both the baby and the clinician. We will also validate the pose estimation metrics that we discussed in Chapter 2. To start, we worked with New York State's Institute for Basic Research in Developmental Disabilities, or IBR, who recruited the infants for us from hospitals and very graciously hosted me and helped me collect the data that we used in the study. For this work, the team recruited infants from the hospital who were both preterm and full-term, and infants whose movement development need to be monitored. So their parents would bring them in for developmental testing, before which I would record the babies with wearable sensors as the clinician performed several tasks with them. So our main goal here was to digitize additional tasks that are not currently able to be recorded through pose estimation means because they would not provide reliable metrics. We also wanted to provide better understanding into the relationship between a clinician and baby during these tests, which is not something that we can currently extract from pose estimation either. So to start, shown here in panel B, we use the APDM Opal sensors to measure gyroscopic data of the wrists, ankles, and torsos of the infants. These sensors come with straps that are suitable for adults, but not for the very delicate and small body parts of a preterm. So we adapted these wrist rattles provided to us from IBR. We cut off the little heads and sewed the bands onto the sensors. On top of this, we had to use athletic pre-wrap to further secure the sensors onto the babies, because once they would start moving, the sensors tended to move along with them. And we know that these were comfortable for the babies because several of them fell asleep after the entire sensor setup, which was inconvenient in the moment, but a true testament to the fact that these sensors did not disturb or bother the babies. So we started off recording the babies in a supine position with the sensors, and we also simultaneously recorded them throughout a phone camera or the Baby Motion Lab app once that was ready. And these videos were processed using the same pose estimation software that we used in study 2. You can see here the sensor placement on the babies is on the wrists, ankles, and torso, and also that we put one sensor on the dominant hand of the clinician. For the supine position, the clinician did not interact with the baby. However, we will see in a bit where that information becomes relevant. We decided to evaluate these 5 postures: supported sitting, pull to sit, supported standing, and supine and prone. These postures have been established as key developmental milestones within the first year of life, and they can provide us with valuable insights into an infant's motor skill proficiency and transitions. They were chosen based on their clinical significance and recommendations from clinicians specializing in the area of the study. So the different postures can allow the clinician to interpret different supports that are building in the infant system by interpreting the engagement of their neck, torso, and the overall movement when in these positions. So in this presentation, we are going to follow just one baby, baby 5, to better appreciate the individualized nature of this analysis. This baby in particular, we recorded for 4 visits, starting at a corrected age of 3 months or 14 weeks, and then monthly until 6 months. The corrected age is the age this baby would have been had she been born on her due date. And it's significant to note that this is a preterm baby who was born about 14 weeks early. So we get a very interesting insight into these early developmental weeks. So here we see data from just visit 1 from the sensor data. We got the gyroscopic time series, which refers to the angular velocity or the rotational speed of the sensors. On the left, you see the raw angular speed from the kinematic data, and we can see the ranges of motion for each limb and the torso. It's clear that the arms display more movement than the torso, which is consistent with what we have found. The pronounced peak we see here in the left leg is most likely an isolated kick, but overall here we can see the importance of the arms, and just in 1 visit you can see that there is quantifiable differences across the body. On the right, we took the micro movement spikes of this raw data, which standardized the angular speed fluctuations based on the empirically estimated gamma mean. As a reminder, the micro movement values range from 0 to 1, with 1 representing activity that strays away from the infant's own resting average. And from this approach, it personalizes the analysis by anchoring it to the infant's unique baseline. This will allow us to compare babies over time, since it adjusts for allometric effects that are inevitable as a baby grows physically, and it maintains the original timings of the raw sensor data, which is relevant especially when interpreting dyadic interactions. So based on the micromovement data, we see a more periodic pattern in the arms, with cycles of small and large deviations from the average, which suggests more active movements than in the leg and torso. The right arm seems to have more significant fluctuations, which hints that at this point there is a slight right arm dominance. So here we track the evolution of the shape and scale gamma parameters over the course of the 4 visits. From visit 1 to visit 4, we see a very significant shift from a heavy tail distribution to a more Gaussian distribution. This indicates a move from less predictable variable movements to increased movement regularity, if you recall from our quadrant divisions. Between the 1st and 4th visits, there is a very clear pattern of development that matches the already established trajectories of a full-term infant, which is interesting considering the baby was born 14 weeks early. This suggests that there is a universal milestone in the noise and shape distributions in motor control during infancy, and with more data, it could help us empirically establish concrete transitional periods and typical development. Here we see the probability distributions from the empirical shape and scale parameters across the 4 visits, so we can visualize the evolution of these over time. We can see that initially the visit 1 distributions are skewed with a higher index of dispersion, and that over time these distributions evolve to become more symmetric, which reflects a higher shape and lower noise value, which again corresponds with what we would expect in a typical full-term infant. And this is a characterization of general movements that also matches what we found with the videos. Now, looking at the data in a higher dimension, we are looking at the gamma moments, other moments of the gamma moment space. So here we see the mean variance and skewness from the infant's micromovement spike angular speed data. We can also see the kurtosis, which is a function of the size of the marker. So the color of each marker represents the overall range of motion from the sensors, with the lower values depicting slower and more controlled movements, and the higher values faster movements. We again see a developmental trajectory from visit 1 to visit 4, the markers for which we can identify based on the edge color. And this trajectory suggests maturation in motor control that we have seen so far, from sporadic and less controlled motions to more deliberate and refined motions. So to evaluate the relationship between the clinician and the infant, we needed to evaluate the temporal synchronization of the kinematic data between each of their sensors. So to do this, we utilize the cross-coherence and transfer entropy data from both of the babies and from both the baby and the clinician sensors to create a weighted directed network that can depict the changes in motor independence across visits. In these network graphs, the node size is the out degree, which is essentially the extent of influence that the particular body part has over others in terms of movement information flow. The thickness of the arrow refers to the transfer entropy value, which designates a directional flow of information. So thicker values have a higher directional influence than thinner arrows. And in some cases, these nodes are different colors, which illustrates the existence of separate modules defined by high connectivity within the module and low connectivity outside of the module, signifying coordinated movement patterns. And this was done using the brain connectivity toolbox, which is typically utilized for brain data, but we utilized it more for physiological data. So these graphs are a visual for us to see the changing dynamics between the clinician and the baby during the pull to sit task. Across the 4 visits, in this task, the baby starts out on their back and the clinician will pull the baby up to a sitting position using their arms and then keep them in a sitting position by supporting their lower back and torso. The amount of support they provide depends on what they gauge the baby needs to stay in that position. The top row of graphs is the clinician's role in the network, which we can see by the out degree arrows from the diamond that represents the wrist sensor on the clinician, and it is essentially how much the clinician influences the infant. The thickness of the lines again reflect the transfer entropy, with thicker lines meaning that the node has a higher or lower degree of influence. Over the course of the 5 visits, we see the clinician's line become thinner and thinner, which shows a decrease on the clinician's direct influence on the infant's movements. The bottom row focuses on the right arm of the infant, and here we see its relationship with the clinician and the other body parts. We see a smaller reliance on the clinician over time, and overall we see more independence within the baby by the 4th visit. The changes in the network connectivity suggests that as the infant grows, and what we can see by the 4th week, they are able to develop their own motor strategies for these tasks and work more independently. The last result we are going to go over is a cross-coherence analysis of the sensor data and the pose estimation data recorded of the baby while they were in the supine position. Here we see a strong congruence between the pose estimation sensor data shown by the high values of coherence, which validates us using pose estimation data with at least a 75% confidence. So this supports the work that we did in Chapter 2 is reliable, and that future work can use this pose estimation toolkit to analyze videos of infants. So overall in this chapter, we were able to validate the pose estimation data, ensuring that we were accurately capturing infants' movements. We were able to track the development of motor trajectories over the course of the 4 visits by looking at the micromovement gamma parameters and seeing the shifts that occurred toward a more predictable regime, and capture that by visit 4, the baby was able to shift toward intentional movements. We also captured variability across the different body parts and validated the order of maturation we established in study 2. And we went past just intrinsic motor development in the infant and were able to include support provided by the clinician and quantifiably measure how the infant gained independence over time by using the dyadic data between the pair. In the future, I hope we can build upon these findings and create reliable tools that clinicians can use in the office to complement their work with more objective metrics, and maybe even deploy such metrics in the homes of parents so they have more frequent and quantitative tracking of motor development. And so in all, this work aims to propose new analytics that are scalable and accessible to clinicians and parents who spend the most time with infants. We introduced the implementation of a home-based monitoring system for more frequent data capture that can lead to the adoption of more scalable technologies. And we introduced various methods that have the potential to track and monitor the development of motor control over time based on just the infant and their interactions with others. To end this presentation, I'd like to go over the main points I believe this work addresses and why it's important. One, we address the need for non-invasive techniques of data collection to accommodate the delicate nature of preterm infants. Two, we address the need for frequent monitoring in a natural setting. We also address the need for scalable solutions that are embedded in accessible technologies, making developmental monitoring more accessible and inclusive. We also address the need for data acquisition, analysis, and pipelines that can be used by clinicians, researchers, and parents together. We discuss analytic methods that are sensitive to the early signs of developmental deviations, allowing for earlier interventions. We also discuss metrics that address the nonlinear and non-uniform rate of growth of the body during infancy. We address the stochastic shifts in both physical growth and motor control that are currently confounded in growth charts, but can be evaluated separately. We provide a digital characterization of the general movement assessment that can aid clinicians in their practices. And lastly, we use a personalized unifying framework for monitoring and tracking change over time to assess not only the child, but the child in relation to their environment and those around them. These are some of my references, and I would like to thank my advisor and my lab mates for all their support, the Nancy Laurie Marks Family Foundation for funding, of course, IBR for all of their help in recording and recruiting, and my friends and family for their support the past 5 years.
Okay, very, very, very well. So let's then, thank you for a wonderful presentation. Let's have some questions from the audience. Any questions?
I think they're speechless, Liz. The motor system is overwhelmed. They can't talk.
It's truly a extremely impressive body of work. I can't believe. I remember during the proposal, we're like, Richa, you're signing up for so much work yourself for like a year or so, and you've done it, and that's, I have to say, huge congratulations, and so impressive, and your presentation was fabulous. I do have a couple questions. Part of it is because of my interest in cognitive development, and I'm in general more curious about like the implications of this body of work. So I have some higher level questions. Like one thought I had was, you mentioned that contrary to the common belief that babies respond to moms' reactions, you found that it's the moms that's more likely to be responding to babies. So, and you found some individual differences in how responsive the moms are to the babies potentially. So what do you think it would mean for the moms to be more or less responsive to the babies, or maybe some cases the babies are also responding to the mom's leads? What would that mean?
Yeah, I definitely think that there would be inter-individual variability between different pairs. But I think ideally what we can take away from this is that normally we use the mother as sort of the foundational base, and we sort of need to shift the lens to see that mothers are also people in this relationship who can be influenced by their infants. And understand that the mother's state maybe needs to be even more regulated on a cognitive level, and understand that their system is more influenced by their baby to help them regulate, and then maybe re-regulate their baby as a result. So I definitely think that it's a loop, that the mom will influence the baby and the baby will influence the mom. But based on what we're finding, it seems like the most direct translation is that a infant's higher state will influence the mom. So I think implication-wise, moms would need to be a bit more aware maybe when their babies might be dysregulating them to help themselves cope, especially postpartum, because that's a very intense period.
Yeah, as someone that's recently been through it, it's definitely, it's really, I think it's really interesting to think about, like what you said, maybe, and it would be interesting to longitudinally track well, if you have more data from these dyads, right, like to know what these differences between the mom's responses correspond to all these other metrics that you got.
Definitely.
It's super interesting. I have another question if other people.
Quick question, Liz. Do you want to do committee questions in the second phase or after
So
the audience, or do we just?
we can do them now and then get into the more technical thing in a second phase, maybe in a like we did yesterday with Mona in a what is it called, a chat room,
A
or
breakout room.
breakout room, yeah.
Breakout room. Okay.
Yeah. And then, so Richa, you could take us to there
Okay.
later when we were, but I mean, general questions, then yes, please, by all means, because that way the audience can also appreciate, what's being presented.
All right, I'll keep asking then. It's extremely interesting, and I remember when I was at your proposal, I was thinking to myself, like, if I ever get a chance, I would record my own baby from day one, and of course it didn't happen.
Yeah, it's
Well,
easier than that.
But so for the last study, it's again super impressive, and I can't believe you got all the data. And maybe you already looked at this. So you showed us that there were developmental changes in the baby's motor patterns, and there's changes in how the clinician interacts with the baby over time. So were there a relationship between these two? Like, so was there direct links between how the baby's motor patterns were developing and how the clinicians are interacting with them?
Yeah, there was. So we can see that by visit 4, the baby's system is essentially going toward a more independent state. And then we see this directly with the clinician, that when the clinician doesn't need to give the baby more support, that it essentially means that the baby's state is more independent. So it's kind of the same metric measured in 2 different ways, but we're able to use the clinician as sort of a proxy of telling us that, see, the baby is stabilizing itself, and it's not just the results that we're seeing when we're looking at the baby independently.
Yeah, and do you see that the individual differences relate to each other? So like how much the baby is independent relates to how much less the clinician is responding to them.
Oh, we didn't look to that degree. That is something that I would need to double check actually, if they're directly linked, but I would assume so, that as a baby basically creates more autonomy in its own system, it's able to stabilize itself better, but it may be task dependent depending on what is more stabilized during the developmental phase.
Yeah, I would be super curious to know.
Yeah, I will look into it and get back to you.
Thank you.
Richa, I have a bunch of questions too, and of course some comments. I'm happy to make them now. I'm happy to wait until maybe general audience members have a chance to ask. You let me know.
I'm not sure if there are any questions from the general audience.
Fair enough. Fair enough.
If-
So first, I'd like to echo Jenny's congratulations. This is really, every time I attend a presentation, a talk by someone from the lab, Liz's lab, your lab, be it Theo or Mona yesterday or you today, I'm really blown away by the sophistication of the work, the novelty of the work. And also one thing that I find very impressive, it's how counterintuitive it is. One would never imagine, when you think casually about it, that so much information with such power and predictive power could be extracted from something like heartbeats or, movements. This, to the naked eye, seems either, irrelevant or completely chaotic. And so I'm going to ask you later what you think about the relationship between this and cognitive science in general. That seems to completely, for the most part, from what I've seen, ignore this kind of stuff, which is so rich and I think so important with its implications. So congratulations, and the presentation itself was really superb, well organized. As I told you this morning, before everybody arrived, I think that on top of all that stuff, you write beautifully well, which made reading the thesis a pleasure. So with that said, I think I have some general questions and I have some more specific questions, but one general question or line of thinking that occurred to me this morning when you were presenting transitions in the baby's development between writhing, fidgeting, and what you call intentionality, is all of a sudden it occurred to me that you have a method to track something that I don't think anybody's been able to even begin to think about, which is the development of agency. So, I mean, as Liz knows, I've been thinking about agency for a while now. I've been doing some work on agency with Laura, who just graduated, but this offers completely new windows. Not just looking at agency in adults, but also looking at how the system develops what we call agency. So I was wondering if you could tell me a bit more about how you would characterize, I've seen what you said, but I'd like you to bring it together, how you would characterize, in terms of all the sophisticated measures that you've deployed, that transition in an infant from something that seems completely chaotic and random to what you call intentional or intentionality. So what changes and what happens, you think, to get to that stage?
So I think the biggest contribution that we have to the development of agency is setting a proper scaffolding system for the baby to explore its environment, which I believe is the direct link to the development of agency. So once a baby can explore and interact with their environment fully autonomously, I think that is basically the precursor to the development of an infant's agency. So that progression toward the intentional movements, before which all of these systems need to be in place. If we can get to that point, I think we can say that we have the components, hopefully. Obviously, right now, we can't say fully whether this would lead to agency or not, but that intentional period and whether the baby is in the right regime during that period would be a good marker for the.
So, yeah, and could you say a bit more about what characterizes, from the perspective of the measures that you use in, what makes what you call intentional movements on the part of the infants? What makes these movements different from the writhing or the fidgeting movements? Are they simply less chaotic? Are they like you have such rich data? I'm sure you can say a lot about the signature of these movements.
They're no longer random. So there is a predictability to it that we can deem that these movements are intentional, meaning the infant is intentionally interacting with their environment. They're intentionally exploring it in a way that they are curious and developing that sense of basically exploration, which is the foundation of agency during infancy. So I think the fact that we're measuring these movements in a way that we can see that based on the infant's own patterns from birth, these movements are now less variable than they were. That progression we can determine is the entrance into intentional movements.
And you also said that there was a relationship between, so you looked at the proximal, articulated, and distal systems of joints and body parts. And remind me again what you found in terms of how these different systems reach that level of smoothness and less randomness, what you call agency. So how does that happen again in terms of over time?
So we see that in the proximal body parts, there is a stability throughout the periods. In the fidgeting, which is very interesting, we see variability in the or first stage, the writhing phase, and again in the intentional phase and more stability in the fidgeting stage in the middle. So this period, I think, is basically a precursor or a very developmental period. There are some underlying mechanisms at play here, which then lead the articulated phase to again have variability during intentional, because it would be different if it was stable from the beginning and then led to a more variable regime. But the fact that we see variability, it disappears and then it comes back.
Yeah, that's fascinating, really. Yeah. So
And that's creating, there are some underlying mechanisms that with more research, hopefully we could uncover.
Yeah. Any ideas about why things unfold the way they do in terms of these different systems of joints, and why you see that variability kind of be there, go away, come back? Or is it something that you haven't begun to think about yet because you focused on so many other things? I'm just curious.
I mean, I think on a surface level, it again ties to the exploration, and that the infant, it wants to explore, and to explore, it needs the development of its distal joints. So the other parts do need to basically go to a structured and predictable area for the distal joints to have that stability. But it is definitely something I need to think about more.
Oh, I mean, there's so many things. So to follow up on this paper that I was looking at, again, by Liz and G of, I think, 2020, where they looked at these really interesting signatures of fully intentional movements, instructed like point there, and then when you retract, and you find that you have different signatures. This was done with adults, I believe, right, Liz? Yeah. I'm wondering if
undergraduates, yeah.
Okay, undergrad, so babies in a sense, but not really, not really infants, so adults. So you can see where I'm going with this. So this was a really important discovery about agency and certain metrics being able to track levels of intentionality in ways that you could not do with any other techniques. Do you think that there would be a way somehow to see when and how these distinctions fall in place much earlier on in life, like say with infants, for example? I mean, do you think that with the technology that you have currently, that you've deployed, do you think there'd be a way to look at this, or would you need something? Would you need to adapt the paradigm that G and Liz used to, which is hard with infants? You can't tell them to, right? That's the thing. But what would be amazing, and one thing that's amazing about your work, is to find all these measures that bypass explicit verbal instructions and stuff like that. So do you think there could be a way to find out how this works in infants, and when you get to these differentiations within the motor system and the nervous system?
Yeah, well, so the work that you're referencing specifically, that provides us with a metric of what we can deem to be intentional movements.
Right,
So
exactly. Yeah.
see that when the baby's movements, even though that they are not goal-directed in the sense that they're not instructed, we can understand that they are goal-directed when the quality of the movements shift toward the more predictable regime. So we can interpret those movements as intentional movements. And when we see that during an intentional phase, we are looking at this intentional metric that we've already developed, we can start to assume that those are goal-directed movements and that there is that development of intentionality.
Yeah, that's
all building off of that prior work.
All building off of what you said?
Off of this prior work and basically quantifying intentionality and agency. Otherwise, we would have no metric of understanding what is intentionality in an infant, because we can't ask them, we can't direct them.
Right, that's the thing. That's what makes those techniques so interesting and powerful, because you can ask developmental questions about things like agency, which, I mean, and go very, very young.
Yeah.
Before they can talk, before they can actually do so many things. So that's really, I have other things, but I'll pause to see if other people have questions and comments. Thank you so much, Richa, for your answers.
Of course.
I would like to again, big congratulations to you, Richa.
Thank you.
It's really fascinating work. Although part of the motor study. We've been working very closely hand in hand, but still like just in a very short period of time, we've done so much work. It's amazing, and the results really like fascinating and very impressive, and kind of like unbelievable that that it could have all those work done and the writing in such a limited time period.
Thank you.
Yeah, so again, another sort of like a comment on the heart rate variability data and the diet between mom and baby. So just keep in mind that the age of the baby here is 12 months.
Yeah.
So yeah, so at a very early age, like you're newborn, you can see that mom's very attuned to the baby's need. So sort of like baby leads during that time period is understandable, but your results, on the other hand, also seems that that's still like a sort of the interaction is still there later on. So it's surprising, but it also is an important finding, and hope that future work kind of like confirm and support this finding. And also it's interesting to say to see that you continue the interaction between the baby and another either mom, another person, either mom or the clinician during the either just the autonomic or it's more voluntary type of system, the motor. So again, like it's and so much information that you can extract from that. So I'm just curious to see like just so how you also like show clear future direction of the work, and I'm curious to see like for your personal future goals, the near future, what would you like to focus on?
I think one of the biggest things I would like to have more data to validate a lot of these findings and over a longer period of time. I think the intentional period is one of the most informative periods, and we see the transition to more better quality of these spontaneous of movements in babies during that time. And unfortunately, I wasn't able to get that much data during then. So I would be very interested to see what patterns and transitions happen there. If there is what the variability looks like at that point, and what we can learn from that period.
Great, and also like you're going to work in a like very top rated rehabilitation facility. So what would you speculate on the time period that early intervention can impact the most? I mean, just like within the timeline that you got like detailed data and analyses.
I think it would depend on each baby when we detect that there's a certain system that isn't developing in the proper way. Because every baby's, obviously, if there's some atypical development, it's unique to each individual. And I don't think that intervention should ever occur if it's not entirely necessary. So if we're able to basically isolate what parts are not developing in the ideal way, whatever the time is that we can earliest detect that, that would be the ideal time for intervention.
Yeah, so again, like the importance of individualized and precision medicine.
Yeah, exactly.
Great, thank you.
Thank you.
So maybe we move to the breakout room if nobody else has questions. Thank you, Richa, for answering all the questions, and thanks to the panel for such important questions and interesting observations. So if no one else has a question or a comment, it could be just a comment. I see some in the chat. Thank you, Jen, for your wonderful comment. You always hit the nail on the head. And like Jen says, this changes the world. Research like this changes the world, and I agree. I concur. The scalability of it is it's a key element here. So if no one else has any questions or comments, then Richa, can you